Right. Hi, welcome to Exploring Light, Week 1 on Perception. This is our fireside chat. I'm Eric Muller, we're one of your instructors here with my colleagues.
I'm Paul Verdi.
I am Zeke.
So this video that you're watching, we're going to answer some of the questions we found online that you've wrote in the discussion sections. And by the way, I just wanted to say I was really impressed with all the people responding and all the input we got from people out there. Hi, people out there. And we're going to try answering those, and we're going to try to go above and beyond that and actually show you things that you might want to try if you haven't tried it already. And before we get started on answering questions, one thing that came up a few times was the pre-course quiz. Some people were having problems with it. And first, I just wanted to say we are sorry. It's just technically that's just the way it is. We had to put this quiz up to get information from you, and unfortunately, it was kind of registered as a graded quiz. And I know that some people that were answering these questions about themselves, maybe they didn't pass, but we're sorry about that. It's just the way it is with the Coursera format right now. But just keep carrying on, and hopefully everything's going to work out great with the rest of the course. And if you have any other technical questions or any questions in general, if you are on live right now, why don't you try sending us those bits of text to us, and let's see if we get anything from people out there in virtual land. And we have 4 viewers right now, but the numbers might climb. If you got something to say or you got a question, just send it off to us, okay? The first thing I did want to talk about was peripheral vision, and I did want to say I was really impressed with all the data that people put in there and all the questions that people were pursuing on that one. But one of the questions that came out, and I'm actually going to look and see who asked this first one over here. This was from, do we have it over here? Yeah, so
From Romania.
Oh, hi, Romania. Was from interested about the distribution of and codes. Is from Martin, and I just wanted to say, because Martin was mentioning that they were interested in knowing why his students could only read text at a certain angle. If you saw the video on peripheral vision, you might have seen this document. This document is for you to download also, but this document is a cross-section of the backside of your eye, and it actually has in it, it has pictures of rods and cones, and so the cones would be in this one, the green blotches, and the rods would be the red blotches over there. But you can notice from the graph over here that the population or the density of rods starts out low and zooms up really quickly. It falls off, and right in the center of your eye, it's loaded with cones right over there. And you don't have nearly as many cones on the periphery of your eye over here. It's mostly rods until you get to the very, very center. So it turns out, and I mentioned this in the video, that the center of your eye is called the fovea, and it is loaded with cones, millions and millions, about 6 or 7 million cones, and that is where your clearest vision is. But it also turns out that it is, I guess, really expensive biologically to have a whole bunch of cones. So what we have done biologically, it just packed the very center with cones and made everything else up, because the sides of your vision is not nearly as clear, even though it looks clear. The only place it's clear is in the very, very center. So that's the only place we can really see the text clearly and interpret it. Off to the side, you can't see it. And when you do that investigation, we're actually looking at the angles of where you see stuff clearly. It turns out it's only a few degrees of clear vision. That's because when you're looking at that clear center right there, that's where the cones are totally packed in and things are very discrete in that area. You have a much better ratio of the signal from the cones going to your brain than off to the sides. Things are bundled differently as you go out here. Over here you have
The eye is part of the brain and is processing the signal on the way in. And the other thing that I looked at, there's a question from one of Martin's students, is that as you did the peripheral vision activity, you brought the object in from the side, somewhere out there around 40 degrees, you could begin to see color. And the graph that Eric just showed indicated that there were no cones out there at 40 degrees. But what you really need to have is a logarithmic graph that shows you the number of cones for real. So I have that on my screen, and I'd like to share that with you. So if you look down at the bottom of the Hangouts and you see the name Paul, if you click on Paul, it should bring up this graph. And this one is done a little bit better. And on this one you can see that the number of cones drops off quickly, around 10 degrees, or a few degrees, but that it does not go to zero. There are some cones out there on the periphery. And that when they get triggered, when they fire, when they generate an action potential, that's when you can see color. And the thing is, the number is small out there, so you're not very sensitive to color. That's the kind of thing you're finding out in this peripheral vision activity.
Yeah.
Okay, so now you go back to the main screen, and do we have questions?
Yeah. So, Luliana?
Sheila.
Oh, where's that?
Sheila.
Oh, this is from Sheila. It says, does the analysis of rods and cones explain why we don't see bright colors, in aurora borealis, but cameras can capture? If so, how does it explain some people able to see pink and greens, but most of the adults saw grayish, bluish colors only?
Yeah, so the colors of the aurora, they're charged particles in the Earth's magnetic field hitting the upper atmosphere and exciting the molecules. And each different molecule up there has different colors it gives off. Oxygen can be green or red, and nitrogen is more of a salmon color. And so you have different color sources depending on the energies of the charged particles. There are two different excitation levels for the oxygen. One of them's red and one of them's green. So that controls the origin of the colors, and of course the salmon is kind of a gentle colored salmon, very often appears white. But now you're looking at it with the rods and cones in your eye. And if the light on the aurora is dim, and it's not enough light to trigger the cones, then it will appear purely as a black and white phenomena. You won't see much color at all. It'll just be that whitish glow. But as it gets brighter, then it can trigger the cones and you'll get some color. Now there are lots of reasons that some people might have different color visions. As you get older, the lens in the eye becomes brunescent. That means brownish. And so older people tend to lose their sensitivity to the blues and the violets. They get filtered out. Mm-hmm. So depending on the age of the person, two people can look at the exact same aurora and see different colors.
And well, there's another factor as well. You're probably going to say this, but your pupil, how big your pupil get also decreases with age. So kids can have a much larger pupil, and so they can gather more light than someone who is older. Right. And so things literally look brighter at night to younger people because their pupil can get larger.
That's why you should never play tennis with a young person at sunset. They can see the ball and you can't. Okay.
I'm just going to mention looking at stars while you're looking at the aurora.
Oh, yeah.
I'm going to just show this real quick. So over here, and then I'll send it off to Paul, but if you notice, right over here is the fovea where your cones are highly packed, but you'll notice off to the sides over here, this is where your rods are really at their peak density. You want to talk about how seeing a star at night relates to this chart over here.
Yeah, if you have a dim star, like if you're looking at the middle star in the handle of the Big Dipper, okay, that's called Mizar. And as you're looking at Mizar, it has a dim companion. And if you look at Mizar, you cannot see that dim companion. It's too dim to trigger the cones, which are the only things in the fovea, which is where you're looking. But if you look to the side a little bit, then the image falls away from the fovea where there are rods, mm-hmm. and you can see the dim companion to Mizar. So it's called averted vision, mm-hmm. looking to the side to see dim things in mm-hmm. the sky.
And that's because the rods are stimulators to few photons, right. as opposed to the fovea and the cones need thousands
That's
of photons to get them simulated up and running.
Yeah.
Yeah. So you could see dimmer things by looking off to the side. Great. One of the things we wanted to talk about is how clever you people are out there. I wanted to bring up questions about colored shadows. People were trying to figure out how to do this colored shadow activity. A lot of people went to the hardware store and bought the bulbs for it, but some people couldn't find the bulbs. They actually had to come up and jerry-rig their own system in order to see colored shadows. And I was going to bring up what one of the participants showed us, which I thought was really clever. And right over here. Did you guys see this?
Yeah.
Yeah. What do you think?
I thought it was amazing. A teacher using filters that she had around the house and a blue jar that had like cold cream in it.
Yeah.
For the blue filter, I thought that's our kind
clever.
of teacher-institute teacher using everyday materials to find a red, a green, and a blue color.
Yeah.
For the white lights that they could find.
Yeah. I don't know what kind of filtering material they have below it, but that jar blew me away.
Jar was excellent.
Yeah, jar was great. So you want to talk about rainbows?
Sure.
Okay, let me hop back on to rainbows over here and stop sharing the screen, and so you don't have to look into infinity over here. Hey, so we had a whole bunch of people writing in about making rainbows for their students in their classroom and putting them on the floor. And Zeke and I and Paul, we talked about it, and we talked about, and we're going to show you how to make them and what you would need to get and do if you want to do this in your classroom.
So the thing you'll want to get is some diffraction grating. And it doesn't look that exciting when you just sort of hold it up. It looks like a relatively clear piece of plastic. But when you shine light through it, I'll get it in the flashlight. You might be able to see.
No, kind of bright.
A little too much. Well, we'll show you it another way.
Yeah.
A diffraction grating makes rainbows. And let me show you how that would work on this overhead projector. So this is like super old school technology, but you'll see what we're talking about. So I'm going to need to turn off some more of the lights in the room, but let's first show you the overhead projector before I do that. So this is it's got a flat bed here where the light comes through. It's going to blow out the screen now, so you put transparencies there, and we'll cover a slit. You'll understand by that in a second. And then it's got this mirror and a set of lenses here, and then it projects that on the wall behind us. Eric, you'll just turn right over there so you can project it. And when you turn off the lights, you can see it clearly. That's the projection on the wall. But what a diffraction grating does is that it takes that projected light and it splits it up. It takes a portion of that light and it splits it up. And you'll see that there's this rainbow over here from it. And on your screen, because of the nature of how screens make colored images and the way the camera accepts colored images, you'll see a little bit later on, that doesn't look quite as smooth. It looks like sort of there are bands of color there. But if you were here, you would see that this is a completely smooth set of colors. There's like no, like when kids draw rainbows, they often draw like a purple band and a blue band, and that's not what it actually looks like in person. It just looks like this smooth color gradient all the way across. And you can make the diffraction grating, there are a variety of companies that make them. One person who makes a very inexpensive one is called Project Star. They sell very nice diffraction gratings. You buy 100 or 200 square centimeters of it for about $15, and you can cut that up into very small pieces. We're using a relatively large piece of it, this card, to make a big spectrum for the whole room. We have this little slit going on top of the overhead projector, and if I change the size of the slit, you'll actually see the colors start to change. As I widen out the slit, we get more of the middle of the spectrum, so you see more yellow, and more cyan. But if I widen the slit too much, they begin to overlap each other, and you start seeing white in the middle from it. And that's because each bit of the spectrum gets spread out, and the whole spectrum starts to overlap in the middle. And this actually explains what you want to do in your classroom if you want to make big rainbows on the floor like we had. So you want to black out an entire window, except for a small slit, and then in that slit you want to put the diffraction grating. And the narrower the slit you make, the more pure the colors will look in the spectrum, but they'll be dimmer. Where if you make the slit wider, the spectrum on the floor will be brighter, but the colors will start to be smeared together with each other. And we have bunches of them. We have a big skylight here in the Exploratorium, and then we have bunches and bunches of these windows covered up black with just a narrow band of this diffraction grating on top of it. Another spot for diffraction grating is that they sell like rainbow glasses. And some of them, however, are called 2-axis diffraction grating, which this one makes a spectrum here, and then on the other side, and sort of standing in that other spectrum. But then the 2-axis ones make buttons on each side as well as up and down, which sometimes confuses students, but it's not really a big deal. You can still use it to make really beautiful spectrums all over your classroom. Does that answer that question?
Yeah. Can I ask you guys a question about somebody I heard about? His name is Roy G. Biv. What do you guys know about Roy G. Biv?
So we often talk about the spectrum. At least in the United States, there's this mnemonic for remembering the colors of the spectrum: Roy G. Biv, red, orange, yellow, green, blue, indigo, and violet. And Newton came up with that list of the 7 colors when he published the book Optics. And he picked 7 colors out of the spectrum, even though there's actually an infinite number of colors here. There are no bands of colors. He picked the 7 because he thought that there should be the same number of colors as notes in the scale. So there's 7 notes, and he thought that there should be 7 colors. But other countries have different numbers of colors. Some say that there's 5 colors in the spectrum. Some say that there are 13 colors in the spectrum. So like different cultures have different numbers. Of course, there really is an infinite number, and color scientists now use a numerical system that indicates that there's an infinite number of colors.
Mm-hmm. Yeah, indigo is my favorite.
Yes, yeah.
Whatever that is.
Yeah, whatever color indigo is.
Yeah. That's great. Yeah, let's turn that back on. And can we get the new line on? Hey, we actually had somebody write in, and where did that end up going?
Yeah, I saw it.
Yeah.
No, there was a paragraph somebody wrote.
Yeah, someone had a question about green receptors and sensitivity and evolution of green. Do you know anything about that, Paul?
Yeah. So one of the we don't really know what drove the evolution. There are two major theories right now about why the 3 different receptors. And it turns out the mixture of the red, the green, and the blue is really good, especially the green and the red, for looking up in a tree and seeing ripe fruit. And so it was thought originally that the receptors had evolved in some of the primates, which eventually we humans got, in order to be able to look out in the distance and see if fruit was ripe or not to make it worth going and climbing. But some of the more recent theories, and there's no proof for these yet, are that the human eye is exquisitely tuned for the colors of human flesh, that we're really into identifying people from our particular band of people. Mm. And so our red, green, blue is really good about sorting out flesh colors. And so, but they don't really know what the actual driving force was. But most of the older orders of primates, the old world monkeys, have just 2 color receptors. Mm. But our band, determined that people have the 3 color receptors: the red, the green, and the blue. And we don't know why.
That's interesting. Yeah. Great. Hi, Romania, by the way. Oh yeah, and so the person who popped that on, yes, sorry, I tested the delete option.
That's why.
If you want to pop your question back up online, that would be great, and we'll look at it again. Or if you have a different question too. You know what's interesting is that so we have 3 color receptors.
Yeah.
So we have a receptor for red, green, and blue. But another way to talk about it is the wavelength. So we have a longer wavelength, a medium wavelength, and a shorter wavelength. And that's all we have. And when I was showing the exhibit on the floor on rainbows, and Zeke was up there showing his over there, we saw the things that we could see. And they're all within that bandwidth that we could see. But I want to show you an activity that is going to be on our snack page. I think you have a write-up for it, right? It's about infrared?
Yeah.
Yeah, so we can't see infrared. We have not evolved to develop it. Other animals do see infrared, and other animals see other wavelengths too. But the interesting thing is this camera right here can not only see the visible spectrum that we see, but I'm going to zoom in so you can see what I'm going to see. You ready? That's the exhibit right there. That's the activity right there. I'm zooming in using a remote control. And it turns out that this camera right here...
shine it at me, Eric.
Shine it at you?
Shine it, Eric. Go ahead, press the button.
Yeah, can you see that, Paul?
I can't see it.
What about you, Zeke?
No, I can't see anything.
Okay. Is anybody else here a snake? See infrared? So it turns out that lots of digital cameras can pick up wavelengths in the infrared, and you out there in receiving land can see this because the cameras process it and they send it into the visible spectrum where we can see. On my digital, my phone right here, I can shine this into my lens over here on my camera, and I can see it also. So I highly recommend, if you've never done this before, grab your remote for your television at home, shine it into your eye, see if you can see it. Man, I'm guessing that you can't. And then take your phone and shine it into your phone and see if it will go into your camera and get reprocessed into the visible spectrum. I will tell you this, some brands of phones have a infrared filter on it. So you might want to try the backside camera. You might want to try the front side camera and see if one of them will process it better than the others. And also you can borrow other people's cell phones and see if they have the filter or how they process it, the infrared on their camera too. But it's interesting. You can make this invisible wavelength of light become visible.
So another interesting thing that goes along with that is that if you've ever tried to take a video or a picture of a fire or like the burner on your stove or something like that, or if you've seen older videos of chefs in the kitchen, the fire on their stove looks super red and it looks huge, and that's because the cameras that were shooting the video didn't have good infrared filters in them. And so even though the fire wasn't really that large, there was lots of hot air that was next to it that was glowing in the infrared, and the cameras could pick it up. My cell phone camera really has that as a problem, and so if I try to like shoot video of me cooking, it comes out really all screwed up because it's getting all these other colors along with it.
Yeah, actually, human sensitivity does extend a little bit into the infrared, but it's exponentially weaker as you go to longer and longer wavelengths. So, but the wavelengths that are used in the infrared remote control are far enough into the infrared that the human eye can't see them. But actually, if you get a bright enough source of infrared, you can begin to see slightly into the infrared. And if you have your cataract removed and you have a new lens put into your eye, the new lens will let through ultraviolet light. And it shows you that the short wavelength sensor, the blue sensor, can actually detect near ultraviolet light. So we have a wider range possible if it's bright enough in the infrared, or if you get rid of the lens, which absorbs ultraviolet, we can extend our range of vision a little bit.
Yeah.
My father is a crazy guy. He likes to go clubbing, even though he's in his 80s now. And he had cataract surgery, and now he says, like, when he's in a club, he says it's like super bright in there. They have all these ultraviolet lights on inside that most people can't see, but he can see. It's like, oh, it's too bright for me. It's like, no. Who would have known?
If he goes to the club and he's 80, have him wear sunglasses. Just the way it is. Pretend he's the Blues Brothers. He's okay.
I will say one thing. If you do go to a club that has ultraviolet light in it, have a drink of quinine water. That's tonic water. It has a chemical in there that will glow based on the ultraviolet light that's coming down. And so you can...
It's called quinine.
Yeah, quinine. Yeah, there you go.
Yeah, amazing chemical.
And you won't get malaria while you're in the club.
That's right.
Hey, I'm going to ask you a question, Paul, and while you do that, I'm going to get set up for something else. But somebody wrote there are also people who have 4 types of chromatic receptors. Is that true, a tetrachromate?
That's right. So it turns out that those people have a very interesting name. They're called women. So it turns out the recipe for the red detector is on the X chromosome. And recently, as we did scientific studies of the X chromosome, they took the X chromosome from, in the first experiment, they did 256 different cadavers and took out their retinas and ground them up and studied the chemicals in the retinas. And they discovered 80 different subtle sensitivity differences for the red cones, that there are 80 different kinds of red cone, at least. And what this means is that since women have 2 X chromosomes, they have 2 chances to get a different red detector. And men have only 1 X chromosome, so we only have, at most, 1 red detector. One thing this does, of course, is that 10% of the men are red-green colorblind. Because if something goes wrong, Zeke, for example, if something goes wrong with that one recipe on the X, they can't see the red very well. But with women, they have 2 chances, and only 1 or 2% of the women are red-green colorblind because they have that second chance. Well, recently, they did some studies. They're looking at most of the women that do have the 2 different X's, that have the 2 different red cones, don't have them wired up. So they can't, if you show them a test, a color blindness test, they can't see any more red colors than the rest of the population. But recently, there was a thought they found one woman who actually not only had 2 different red detectors, she also was wired up to be able to see subtle different shades of red. But there's still some scientific argument going on about her case. So it's really an interesting question, and I look forward to finding out about the tetrachromats.
Yeah. We have another question out there. This is from Kaylee. It says, that's interesting. There is a theory... Oh, we moved it up. There's a theory that exoplanets that orbit red dwarf stars might have differently pigmented planets. Also, I think for related reasons. Well, we have to find life on other planets to begin with, but...
Yeah, so some algae on Earth are red and use red pigments, and some are blue-green. We have blue-green algae, and they use blue-green. So even on Earth, there are different plant colors, if you will, that bring in sunlight. But yes, I would bet that organisms evolving on a different star system might have different sensitivities.
Yeah, well, just on that note, just animals here on Earth, they have different sensitivities. There is bees can see in the UV, snakes can see in the IR, and there's, what is it, the mantis shrimp? Is that the one? It has, how many different sensors does that one have? 13
or more.
Yeah, something like that.
Yeah, it has a huge amount of sensors. So if we do find life on other planets, depending on the radiation that comes off of that their star, maybe they've evolved a different way to receive their light and process it.
Yeah, some fish have 5 cones. You can imagine when they look at that spectrum, they might see 5 bands across it. So
look at that lure right there.
Yeah.
There's another question over here before we get to my demo over here. It says, and this is for both of you, why does IR radiation make things hot, but higher energy visible light does not? As much anyways. Is it simply because visible light does not excite molecular vibrations in the same way?
So visible light will indeed make you hot if there's enough of it. And it's just that your skin is really good. Your skin is really absorbent for infrared. So how do you detect infrared? Well, you use the back of your hand or you use your cheek. And those are 2 places on the human body that are most sensitive to infrared radiation. In the infrared, they are black. In the visible, mine isn't. And so it's very sensitive to the wide range of infrared energies. So it's just your receptor is better at detecting the energy in that infrared. But if you have visible light with the same energy per square meter, and you put it onto skin, some more of it's reflected, but that visible light can carry thermal energy just as well as the infrared, and ultraviolet can carry thermal energy just as well as the infrared. But in general, the sources that we find out in the world, thank goodness, if the ultraviolet has enough energy to be felt on your skin, your skin cells are being killed. So we tend to avoid places like that.
Very good. I am pulling up in 1 second. I'm going to pull up a snack. So just to let you know, we have many snacks on our website, and it's only going to get bigger. Here is one over here. Let me do the entire screen. I'll share that, and let's look over here. So here's our website over here, and here's a snack that just got on our website just a couple of weeks ago called Pixels, Pictures, and Phones. Just to let you know, the picture of that kid there, it's my kid.
Okay.
And it is a write-up that you might want to look at. It's how to investigate your cell phone or mobile device using a microscope or some other magnifying viewing device. And I'm going to pop back over here to my camera, and I'm going to switch cameras. I actually have this device here. This is, I guess, known as scope on a rope. They come in a variety of brands, but this one you put it on a surface and it'll magnify it. And this one I believe is probably maybe 10 times magnification. And so I'm going to hopefully this will work. I'm going to pop over to this, and let's see what we can see. You can watch my code. Oh, is it coming in?
There you go.
Oh, did we stop? Can we browse?
No, it's on there.
It's on there? Okay, here we go. And I actually,
Oh, wait, you just hold it.
yeah. No, I'm gonna.
There you go.
Yeah. So I actually.
Turn off the
Yeah.
light.
So it turns out that there's. There we
There
go.
we go.
So this is a picture of my iPad, and you can see the pixels, and the pixels are red, green, and blue because that's what we have receptors for. So all images we see on these devices are made with red, green, and blue subpixels to make up one pixel, which is made out of the RGB. This is an iPad, and that's what their displays look like. And over here I have a different kind of device that will present information to me. And this is a Samsung device, and these are little mini LEDs. And you can see this one presents information quite differently as far as the technology goes, but they're still using the same tricks that exploit the fact that we only have receptors for red, green, and blue receptors in our eyeball. I'm actually going to, and you won't be able to see this on. Oh yeah, here's my computer screen. Let's see. This is my Macintosh. That's my Mac, and that's how it's presented. And I'm going to run back. What's that?
This is an older one.
Oh, his screen is older. Actually, his is the same year as mine. But this is, I have it now back on my Samsung phone, and you're looking at white. So the red, green, and blue LEDs are lit up, and it stimulates in my eyeball. It stimulates all three of those colors, sends a signal to my eye, and the weird thing is red, green, and blue tell me I'm looking at white. This is white. I'm going to press different colors on my screen. This one's called Clockwork Orange, and this is how this screen makes that orangey color. Here is like a pink color. That is pink. Here is, this one's called Almighty Blue, virtually all blue. Oh, here's yellow. So talk amongst your neighbors. Think about what you would get when I hit the yellow. Here you go, yellow. Yellow. So yellow is stimulated by your red and your green cone, and we take advantage or technology takes advantage, let me get back to the conference camera right here, of the fact, oh, you can't, oh, that's interesting. Yeah, there we go. So this is yellow right over there. But technology takes advantage of that we can only see those 3 colors. You guys want to add anything to that?
Sounded good to me.
Sounded good to you. Okay. And the colored shadow activity that Paul had a video on and has a write-up on, you can play around with those colored bulbs, and you can see that you can make virtually any color just using those 3 bulbs, red, green, and blue.
One thing I should point out, Eric is using this scope on the rope to do this experiment, but if in your classroom, if you just have a good magnifying glass or a low-power microscope, it'll work just as well. But it's really nice to be able to project it.
Yeah. Also point out that the pixels, the size of the pixels on television screens are much larger than the ones on cell phones. In fact, cell phones and iPads have the smallest pixel size because they're trying to put a lot of information in a small place, and the pixels have to be very small. Computers tend to be larger, and TVs even larger than that. The reason why that's useful is that lets you then you don't have to use as much magnification to look at them. In fact, TVs are so large that if you just put a drop of water on the television, then you can usually see the different colored pixels just in the drop of water.
Yeah, I just want to tell a story that I was teaching a class, this is probably 15, 20 years ago, and I told the class of teachers that all the colors we see are composed of the long, medium, and short wavelength of light, red, green, and blue. And after the class is over, one of my students went home. He got a cold. He sneezed on his computer. And he sprayed his computer with, well, I won't tell you what he sprayed it with, but the little droplets that were on there were just the right shape. He could actually see all 3 colors, red, green, and blue. And he was very excited. He wrote me an email and said, he told me, he said, Eric, you should sneeze on your computer, which, you know. Okay. So we have another activity from Zeke that is not on our list, but it's something you should try. You want to hit that one?
We need the document camera a little bit for it.
I want to have them switch to this one.
Yeah, there we go. Yeah. So can you switch to, what is that camera called? The one with 2 circles. If you want to switch to that one, you can see Zeke.
So I have a marker here, a yellow marker, and I'm going to color in this very simple Venn diagram with this yellow marker. It'll take me just a second to fill it in. And so you'll probably notice that these markers look handmade, and that's because they are. So I've always been annoyed by the choices that Mr. Sketch gives me, and I have made my own markers. So this color is easy to identify. It's yellow. There we go. And then this marker is also relatively easy to identify. It's a bluish color. Yes, everyone. Sorry.
Oh, on the camera, it's almost black.
It doesn't look almost black on the camera. Can you turn on the light there so that we can have more light on it? Is there a way to turn the light on this?
Yeah, I will get the lamp on. Yeah.
There
you go. Yeah, yeah, a lot better.
Who knew? No, this lamp is fine. We'll have to wait for it to recolor adjust that. There it goes.
Can you turn the lamp
Can
on?
you turn the lamp on and off again? No, just.
Yeah, looks about as.
There we go. Okay. So here's this blue marker. And so the question is what color will we get when the blue overlaps with yellow? And what I was taught when I was in elementary school is the color that we would get when they overlapped would be green. But weirdly, that is not the color you get. you get a color that is extraordinarily close to black. And that's a little bit surprising that you get that color. And the reason why you get that is how color markers actually work. So the yellow marker is yellow because it blocks out blue light. It doesn't let blue light go through. It lets the entire rest of the spectrum, except for blue and violet, to go through it. And this blue marker is blue because it blocks the whole spectrum except for blue. And so these two markers together let through essentially no common colors. And so that's why that square looks black. So you're probably now wondering then, if that's true, what colors actually do make green? Well, it turns out it's a color that's quite similar to blue, but not exactly the same color as blue. And that color is called cyan. So that's similar, but not the same as blue. Here, I'll put a little blue next to it so you can see the difference between those two colors. And when we overlap yellow on top of cyan, you can see that then makes green. In fact, we can fill it all the way in, and you'll see this lovely green color where the two overlap. And the reason for that is that cyan is an interesting color. What cyan is that cyan lets through all the blue and green parts of the spectrum, but blocks the red parts, red and orange parts of the spectrum. So yellow blocks blue, and cyan blocks red. And so the part of the spectrum that can get through both of them is green. And if we mix magenta in it as well, then we can get the rest of the colors. So here's magenta, and when it overlaps with yellow, it makes red, sort of a red-orange color. And when it overlaps the cyan, it makes a color quite similar to that blue that we had before. And so this is how your printer makes different colors. And if I actually take the cyan and then put it in this middle spot here, if I take it all the way across, you'll see that it makes a color really close to black, as it blocks all the different parts of the spectrum. So the cyan blocks the cyan blocks the red part of the spectrum. The magenta blocks the green part of the spectrum, and the yellow blocks the blue part of the spectrum. And so together, then they block the entire spectrum. And the thing is, though, is that if you go to the store and you buy markers, they often don't label them for you, cyan, magenta, and yellow. And even when they do, they don't actually necessarily do what we would like them to do. They don't always block out the parts of the spectrum we would like them to block out. So I made my own markers, and the way I made them was using bulk printer ink for inkjet printers. And so you can buy blank markers, and you can use printer ink to inject the colors in, and they come with cyan, magenta, and yellow, and so you can make then colors that actually mix together to make the ones you want, to make the real red and the green and the blue, and then mix all the way together to make black. And this will be up on our Snap page in just in a minute, soon. Great, and it's called Truly Primary Pigments.
It's great. Yeah, and it's pretty too.
Okay, so a bit of language, which we have to get to, and that is when we're talking about the cones and rods and neurons, we use a shorthand language, and we say that those rods and cones fire. And what's happening is that when light strikes a rod or a cone, it will trigger what's called an action potential. But in elementary school or middle school, you don't want to go around saying action potential necessarily, so we use the word fire. And action potential is about 1/10 of a volt signal. So every color, red, green, blue, any combination, is a set of 1/10 of a volt signals in a neuron. They're all 1/10 of a volt, and goes down the optic nerve to the visual cortex, where it's finally interpreted as a color. So we like to say, without a human eye and brain, there is no color. The color is our creation of how we visually represent 1/10 of a volt signals. It's really rather interesting. Now, in the eye, the way it works, in particular, we had a wonderful sharing of a video by Doug Stith of waterfall effect, whereas you go out and you stare at one point in a waterfall, don't follow the water, and watch it for 30 seconds or so, and then you look at the rocks next to the waterfall. The rocks flow upward, or your friend's face will flow upward. And the reason is that the way the neurons work is that when you see something moving upward, a neuron in the retina to detect upward motion fires more rapidly. And as it fires for a long time, 30 seconds more rapidly, it adjusts its sensitivity. It's called adaptation. Now, for elementary school, we say the neuron gets tired. But the actual term is that it adapts to the upward motion. Now, there's another neuron that fires when there's downward motion. And the amazing thing about the brain is when you look at something stationary, both of them fire at a background rate. And so it's saying up and down, up and down, up and down, and the brain interprets that as stationary. But if you tire out or adapt the downward motion one, the waterfall, it's all adapted, and now you look at something stationary, the downward motion one is tired or adapted, and the upward one is still firing at its normal rate. And so your brain says, oh, the upward one is firing more, therefore the rock I'm looking at is moving upward. And this is true throughout a lot of our sensory systems, where instead of having no motion or no signal be zero, it's actually both signals, both antagonistic signals firing together. And the ratio is what gives you your perception.
Mm-hmm. Sounds great.
So that's called the antagonistic neuron system.
Hey, Paul, you want to talk about, you just did a big trip, didn't you? Speaking of light.
I just did a trip to see darkness. There was a total solar eclipse in Indonesia and the Pacific, and I just went to Woolly Eye Atoll, where I was the announcer for the Exploratorium's live coverage of the total solar eclipse. And so if you go to the Exploratorium main website and you type in the search box eclipse, you'll come up and be able to see the 5-minute video of totality.
There it is.
And it's really, quite good. In fact, so go see it. We'll talk about the light and the color from the total solar eclipse.
Yeah, and this is the website right here that we're presenting to you. Definitely look at it. Paul, was it a rough trip out there?
It was a rough trip. We actually were on a luxury yacht going across the Pacific to this remote, wonderful tropical island. I had the best fish I've ever had in my life. We suffer for science, you know.
That's. And one final thing I'm going to say is several people in the Teacher Institute, that's the group that we're in, we're heading off to the National Science Teachers Convention over in Nashville. And if you're out there, there are, we range from 15 to 17 to 18 live viewers, but some of you might be viewing this hopefully in the next couple days or so. If you notice this and you get this message, we're going to be out in Nashville, and we're presenting at several presentations, and several of them that we're presenting are on human perception. So if you want to come out and see us, you could ask us questions live. Not just in this virtual world, but actually in the 3D world out in the NSTA convention.
Don't believe it, Eric doesn't actually exist.
I don't know, really exist.
An AI.
So I think that's it for our fireside chat.
We'll watch for you on the Grand Old Light Opry.
And the next week, what is going on next week? Who is running next week?
I'm running next week, I think, and it's going to be the history of light. We'll go through some of the background discoveries about light, working up to answer one of the big questions that we were asked already, which is, so what is light anyway? Is it a wave? Is it a particle? What is it? And we won't get to the answer to that until the 4th week, or an approximation of the answer of that until the 4th week.
That's great. Yeah, so we have a couple more weeks to go. Hope you stick around. It's been great looking at all your responses, and I hope you're enjoying doing the snack activities and taking this course.
We're enjoying your posts.
Yeah.
So keep asking questions. Keep sharing.
So let me see if I got this. I have to end this the appropriate way. Here we go. That's the end of our fireside chat.